Hello, dear learners. Very good morning. Welcome to my YouTube channel. Today we will discuss another topic of plant physiology, and which is C4 cycle, and it is also known as Helson's-like pathway, after the name of scientist who discovered this and explained this cycle. So, as the Calvin cycle, in which carbon dioxide is fixed in the dark reaction of photosynthesis. And this C4 cycle also operates to fix the atmospheric carbon dioxide, but in different way. We will discuss it today in detail. So the C4 plants are, for example, sugarcane, maize, and sorghum. Sorghum is jowar. So these are the plants in which C4 cycle is operated. These plants are of tropical and subtropical region, which tolerate the high temperature and sunshine. So in the C4 plants, there is a peculiar anatomy is found, which is known as kranz anatomy, and this is the vertical section of leaf of the C4 plant, for example sugarcane or maize. So if we see in the anatomy of the leaf, so here as usual there would be upper epidermis, lower epidermis. This is xylem and phloem. So this is vascular bundle, and outside the vascular bundle, this is bundle sheath cell, the which is sheathing the vascular bundle. Okay, this is bundle sheath cells, and these are the mesophyll cells. Okay, these are the mesophyll cells. These are the mesophyll cells. So the only difference from the C3 plant, mesophyll cells has the chloroplast as usual, but in the bundle sheath chloroplast, bundle sheath chloroplast don't have grana. Means there are thylakoid membranes, but not stacked on one another. As we have seen in the chloroplast, and there is thylakoid membranes. Thylakoids are stacked one over one another to make this pile, but this grana structure is not found in the bundle sheath cells. Okay, this grana structure not found in the bundle sheath cells. So in the bundle sheath, the only thing that happens is the C3 cycle. In bundle sheath cell, C3 cycle happens. The Calvin cycle operates in the stroma of the chloroplast, and in the mesophyll cells there operates the C4 cycle. In the mesophyll cells C4 cycle operates, and in bundle sheath cells C3 cycle or Calvin cycle as usual operates. And now see, this is the scheme. This is a scheme of the C4 cycle, and this is mesophyll cell. Okay, this is mesophyll cell, and this is bundle sheath cell. Okay, so the if we see in the C4 cycle, okay, in the C4 cycle, the carbon dioxide from the air is taken inside the leaf through stomata, and when it comes in the contact of water, it is converted into the bicarbonate, this SCO3 minus, and phosphoenol pyruvate. Phosphoenol pyruvate is a substrate which accepts this bicarbonate to make 4 carbon acid, that is oxaloacetate. You see, phosphoenol pyruvate. This is phosphoenol pyruvate. This is phosphoenol pyruvate. In the ionic form, how you can write it? CO minus. Okay, this is phosphoenol pyruvate, and this carbon dioxide, this bicarbonate, is added to in this reaction catalyzed by the phosphoenol pyruvate carboxylase. Here, it is catalyzed by this enzyme, not by the Rubisco. Okay, and this phosphoenol pyruvate is converted into the oxaloacetate. See, this is oxaloacetate. Hmm. So this carbon dioxide is added here in the form of another carboxylic group. So phosphoenol pyruvate is converted into the oxaloacetate, oxaloacetic acid, and this oxaloacetate and phosphate inorganic, this phosphate is released here in the term in the form of phosphate, inorganic phosphate, and this reaction is catalyzed by the PP carboxylase. Now oxaloacetate is converted into the malate, and the this oxaloacetate is converted this oxaloacetate is converted into the malate. Malic acid, malate. And in this reaction, H hydrogen of the NADPH is added here. Okay, as you can see, in the oxaloacetate there was ketone group, and here hydroxyl group is added, and this reaction is catalyzed by the malate dehydrogenase enzyme. Now this malate enters into the bundle sheath cell through the plasmodesmata. These are the channels. These are the channels called plasmodesmata, and this malate here the decarboxylation reaction happens, and malate is converted into the pyruvate, and this enzyme is catalyzed by the malic enzyme. See, this malic acid or malate is converted into the pyruvate, and in this reaction carbon dioxide is released. So this carbon dioxide is released here. Okay, so malic acid malate is converted back into the pyruvic acid, pyruvate. Okay, and this pyruvate is then converted into the phosphoenol pyruvate, and this cycle is completed. So here the decarboxylation reaction is catalyzed by the malic enzyme, and this carbon dioxide is then enters into the Calvin cycle or C3 cycle, and by the Rubisco enzyme, carbon dioxide is incorporated in the Calvin cycle, and 3-phosphoglycerate is made, and further reaction converts fix the carbon dioxide into the carbohydrate or glucose. So the sugar is made in the C3 cycle. So in overview, what we have seen is in the C4 cycle, phosphoenol pyruvate is the acceptor of carbon dioxide, not the RUBP, not the RUBP as in the case of C3 cycle. In the C3 cycle, RUBP was the acceptor of carbon dioxide, but in the C4 cycle, phosphoenol pyruvate is the acceptor of carbon dioxide, and from the phosphoenol pyruvate, oxaloacetate is made. Oxaloacetate is made, and so we can say the carbon dioxide is released in the bundle sheath chloroplast. Carbon dioxide is released in the bundle sheath chloroplast. Here, carbon dioxide is released in the bundle sheath chloroplast, where in the chloroplast there is no grana, so only stroma is there, and there is viscoenzyme and other enzymes which catalyzes the Calvin cycle. So, in the bundle sheath chloroplast, Calvin cycle or C3 cycle operates, and in the uh, mesophyll cells, C4 cycle operates.